Hi, my name is Kathleen Capaccione. I'm an assistant professor at Columbia University in the Department of Radiology, and I was the recipient of the RSNA Resident Fellow Grant in 2019. The topic of my research is targeted radiotherapy with a specific focus on non-small cell lung cancer. Current treatment modalities involve external radiation. However, these have a lot of dose-limiting side effects, and therefore our research seeks to deliver targeted radiotherapy by attaching a radioactive payload to a specific targeting molecule, which can ultimately reduce those side effects while giving people the benefits of radiation that they have already been receiving. So FAP-targeted radiotherapy, which is what we specifically study, has been shown as effective in other types of cancer, such as pancreatic cancer and osteosarcoma, but never in non-small cell lung cancer. So using the money from the grant, we were able to develop a mouse model in our lab that was syngeneic, meaning that it was from a mouse tumor. This allowed us not only to study the overall results of the anti-cancer therapy, but also to look at the mouse's immune system and how it responded to it so we could understand the mechanism of the response. And so using this model, what we demonstrated was that FAP-targeted radiotherapy is an effective anti-cancer therapy for non-small cell lung cancer. It reduced the growth of the tumors by about half. And when we looked at how this worked in connection or in concert with checkpoint inhibitor immunotherapy, we saw that there was an increase in some of the markers, such as those for apoptosis, and a decrease in cell cycling, which suggests that when used together, it could result in an increase in efficacy of either agent. The funding from the RSNA grant enabled me to conduct this research, and it had a number of important benefits. First was that it allowed me to actually buy the supplies like the cell lines, the mice, the drugs, the radiation therapy, and the targeting molecule, all to actually get the work done. So that was really amazing. But through the process of that, it also gave me the opportunity to learn how to work with radioactivity, how to conjugate molecules with radioactive substances, how to develop my skills in syngeneic mouse models. And so all those things gave me the ability to be an independent investigator in the field of targeted radiotherapy. And so really, in a way, launched my career into this field. So I'm extremely grateful for the grant because of the amazing training that I was able to receive as a result of having that funding available to me. And resulting from that grant, I applied for and was awarded the Columbia University Gerstner Scholars Award, which is an early career faculty grant that will provide substantial funding for the next 3 years for me to start my lab and to continue this radiotherapy research, both increasing the preclinical work, looking deeper into the mechanisms underlying the combined targeted radiotherapy and checkpoint inhibitor immunotherapy response, and then also to translate this into a clinical trial, which is planned, which will look at the imaging, FAP-targeted imaging in conjunction with the therapy and how the therapy can actually change the expression of FAP that we see. My advice for young investigators starting out is to do something that you're passionate about. I've wanted to be a cancer researcher my entire life, and it's something that has, helped me, have a pathway through first college and then medical school and then residency. And I think it was my passion and my true fascination with it that sort of led me to be able to apply for and successfully, be granted this RSNA grant. And so my advice is find something that you love and pursue it passionately. And RSNA is there to support you through that process.